In this section, we are going to be looking at the effect of dilution of the solution of a reducing sugar on the color produced upon reduction of a copper 2-sulfate solution. My name is Lenya Jude Fonba. I am a holder of a Bachelor of Education degree in Life and Earth Sciences from the University of Marua, Cameroon, and a Doctor of Philosophy degree in Clinical Biochemistry from the University of Chang, Cameroon. This experiment is based first on the fact that reducing sugars have free aldehyde or ketone groups that can donate electrons. When heated with Benedict's or Fehling solution, these sugars reduce blue copper 2 ions in the reagent to copper 1 oxide, which forms a colored precipitate. Secondly, the intensity of the color depends on the concentration of reducing sugars. A solution with no reducing sugar produces no color change. A sugar solution of low concentration will give a green color. A moderately concentrated solution will give yellow. A high concentration will give orange, while a very high concentration will give brick red. For the concentrations of glucose in grams per liter that can give these color changes, the blue color is obtained for the solution with no sugar, 1 gram per liter for green, 2 grams per liter for yellow, 3 grams per liter for orange, and 4 grams per liter or higher for a brick red color. These concentrations are obtained by dissolving the various quantities in 1 liter of water. Serial dilution is a stepwise method of reducing the concentration of a solution by a fixed factor in each step. By diluting the sugar solution in a stepwise manner, the concentration decreases progressively, leading to lighter color changes with each dilution. We are going to use a set of materials and solutions, including powdered glucose, which will be used to prepare the stock glucose solution. 4 test tubes will be needed to hold solutions. A test tube rack is needed to hold the test tubes. Syringes will be used to measure volumes of solutions. A test tube holder to hold the test tubes during heating. A boiling water bath for heating the mixtures. A pot and a stove can also serve the purpose. A wall clock with a sweeping second hand for timing. As for the solutions we need are going to use distilled water to prepare the glucose stock solution by dissolving 4 grams of glucose in 1 liter of water. This will be called, say, solution S. We also need Fehling solution A and Fehling solution B. Benedict's solution can be used instead. We will be asked to prepare 100%, 75%, 50%, and 25% dilutions of solution S provided. Start by labeling 4 test tubes A to D to hold the respective solutions, 100% to 25% dilutions. Set a final volume, for example, 2 milliliters, which is the regular volume of the stock solution used in most food tests. Note that the percentage dilution represents the percentage of the final volume that will be occupied by the stock solution S. For example, for a 75% dilution, the volume of solution S will be 75 over 100 times the final volume 2, which is equal to 1.5 milliliters. Then complete the volume with 0.5 milliliter of distilled water. With this in mind, complete the 2 milliliters volume with water and the glucose solution to have the final volume as follows. For the 100%, the solution will be undiluted. The dilution factor here is 1:0. For the 75% dilution of solution S, 1.5 milliliters of solution S will be completed with 0.5 milliliters of distilled water. The dilution factor here is 3:1. For the 50% dilution, 1 milliliter of solution S will be completed with 1 milliliter of distilled water. The dilution factor here is 1:1. For the 25% dilution, 0.5 milliliters of solution S will be completed with 1.5 milliliters of distilled water. The dilution factor here is 1:3. After this, add 1 milliliter each of Fehling solutions, A and B, to the test tubes using a syringe. Mix the solutions gently and place the test tubes in a boiling water bath for 3 minutes while shaking from time to time. You will be required to report how you made the dilutions. A sample report of this procedure would be: a final dilution volume of 2 milliliters was set. 4 test tubes were labeled A to D for the respective dilutions of solution S. Tube A to hold the 100% or undiluted solution. Tube B to hold the 75% diluted solution. Tube C for the 50% dilution. And tube D for the 25% dilution. The volume of solution S in the final volume was calculated by Multiply the percentage by the final volume. Water was used to complete the volume to 2 milliliters. The amounts of solution S and water put in the various tubes were as follows. Tube A had 2 milliliters of solution S and no water. Tube B had 1.5 milliliters of solution S and 0.5 milliliters of water. Tube C had 1 milliliter of solution S and 1 milliliter of water. Tube D had 0.5 milliliters of solution S and 1.5 milliliters of water. We will continue with the report in the... Let us continue with the report of the procedure. The composition of each test tube is shown on the table. Here you then draw the table to show the composition. You may need to continue reporting the procedure. You will progress normally depending on the question as 1 milliliter each of Fichling solutions. A and B were added to the test tubes using a syringe. The test tubes were shaken gently to mix the solutions. The mixtures were placed in boiling water bath for 3 minutes while shaking from time to time. After 3 minutes, the color changes in the various dilutions were as follows. The undiluted 100% solution turned brick red, the 75% dilution turned orange, the 50% dilution turned yellow, and the 25% solution turned green. Looking at the variables in this experiment, the independent variable is the concentration of each diluted solutions. The dependent variable is the intensity of the color produced, that is the color change observed. The controlled variables of the constants are the volume of failing solution added to each test tube and the heating time. As conclusion, we can say, the intensity of color change in the test for reducing sugars depends on the concentration of the solution. Less concentrated solutions give lighter colors, while highly concentrated solutions produce a heavier precipitate. We have come to the end of this presentation. Our next presentation will be on the test for non-reducing sugars. Hope you enjoyed following up the presentation. Please subscribe to this channel, like and share. By subscribing, you will be getting notifications when new presentations are available.